to the next speaker, that is Karolina Świtońska-Kurkowska. Sorry for the pronunciation if it's not correct. So she's a PhD candidate at the Department of Molecular Neurobiology at the Academy of Science, Poznan. So welcome. And she will talk about juvenile HD-fused brain organoids of dorsal and ventral origin to understand the early neurodevelopmental aspect of neurodegenerative disease. And she also has a poster B1 that I invite you to look at. Thanks.
Thank you very much for a nice introduction, and thank you for inviting me here. So the talk of my presentation is juvenile HD-fused forebrain organoids of dorsal and ventral origin to understand early neurodevelopmental aspects of neurodegenerative diseases. The aim of our study is to discover disorders of brain development in Huntington disease using juvenile HD-iPS cell lines and fused dorsoventral forebrain organoids. So basically, we use several different iPS cell lines, either HD or control lines, and we differentiate them to a study of neurospheres, so non-adherent neural stem cells, and by adding different previously established small molecules, we differentiate them to dorsal cortical organoids and to ventral striatal organoids. And then at the day 16 of our protocol, we fuse them together in a droplet of matrigel and differentiate them till the day 60 of our protocol. And next, we do several high throughput and low throughput analysis. First of all, we did some molecular characteristic on our 60-day fused organoids and also on those not fused to see whether we have a proper expression of dorsal or ventral markers. And also on the picture on the right side of my slide is the slice of a fused organoid in which you can see the dorsal part because of the robust expression of TBR1 cortical marker. We checked several other markers and we show successful differentiation into mature neurons and specification into various distinct forebrain regions, such as ganglionic eminence, choroid plexus, oligodendrocytes, and subventricular zone and ventricular zone of the cortex. Via bioinformatic analysis of available transcriptomic data, we selected a series of markers which show altered expression in our fused 60-day-old juvenile HD-fused brain organoids. As for our high-throughput analysis, we did single-cell RNA sequencing on 2 control lines and 2 HD lines with 71 and 77 CAG repeats, and also on mosaic organoids, which we did by fusing the dorsal part from the control line and the ventral part from the HD line, and the other way around, of course. And on this UMAP graph, you can see the nice set of juvenile HD organoid cells, as well as control and mosaic organoid cells. Thanks to the specific algorithm, we found around 200 cell subtypes in all of our analyzed lines, and we compared them with the known markers of the developing human brain. And thanks to that, we were able to reveal distinct neocortical subpopulation which is affected in HD and exists only in our HD fused brain organoids, and also in mosaic organoids, but only in those in which the dorsal, so the cortical part, comes from the HD line. To conclude, we successfully differentiated juvenile and ultra-juvenile HD iPS cell lines to 60-day-old fused dorsoventral forebrain organoids. The differentiation and proliferation is visibly quicker in HD fused brain organoids. A series of markers of different neuronal lineages shows altered expression in HD brain organoids. And by high-resolution single-cell RNA sequencing, we revealed distinct neocortical subpopulation affected both in HD fused organoids and in mosaic organoids in which the dorsal part comes from HD line. Thank you very much for attention. Thank you. Many thanks to my department, especially to my PI, Professor Maciej Figiel, and to all of the members from my team. Thank you very much.
So many thanks, Karolina, for your talk. Very interesting. So any questions from the audience? Yeah,
Hi.
thank you.
Thanks. I think it's a really interesting talk. I have actually two questions, if I may.
Sure.
So the first one to me was, did you also add or mesodermal cell lineage in your model, especially for the blood vessels, for instance?
No, we didn't do that. We know that it would probably it would result in maybe better results actually, or even longer differentiation, but we didn't do that yet.
Okay. All right. And the other question was about your single cell sequencing, because I think you're still working at it, but do you also want to look into the glial cells, especially from the dorsal and the ventral part of your organoid, and compare them?
We actually are in the progress in analyzing all of our data, but yes, probably we will do it.
Okay.
Yeah. Thank you.
Cool.
Thanks. Other questions? Yeah. Okay.
Hello. Very exciting and interesting work. I just want to know, the mosaic organoids that you developed, it was fused on day 60, is it?
On day 16, yes.
How long did you grow it, extend it?
Till day 60.
After that, you just took it for the analysis?
Yeah, we just established the time point, and it was day 60 because till that time we had the best result in differentiation, maturation of our organoids. Yes.
Would you like to consider to extend for some more days and see if there is any from the mutant, the HD to the normal organoid, normal organoid, there is any transportation of this mutant, something like that? We see, intracellular, we know it, we see the intracellular movement of, we know the prion-like actions happening in this kind of disorders. So just was curious to know that.
Could you maybe rephrase, because I just don't maybe get the question.
It's like you fused normal as well as HD organoids, right? So you saw only the HD organoids having the HD kind of phenotype, and normal was normal. That's what you mentioned, right, in the organoids? Okay, maybe on the poster I can talk to you in detail.
Thanks.
Yeah, I invite you to my poster. Maybe we can just,
you can discuss maybe later, because we are a little bit running out of time. Sorry. So thanks again, Karolina.
Thank you very much.